Let's take a look at Pablo the penguin from the Backyardigans and why his color is blue. Like all traits, they are formed by the process of creating genes from DNA through proteins. This begins with the process of transcription and can be found in the nucleus of the cell. Transcription is the process of making an RNA copy from a DNA template, and all forms of RNA can be made in this way. After this, the RNA can be made into proteins for further use. However, the entire process has 3 steps: initiation, elongation, and termination. mRNA is created first. mRNA is the monomer of a nucleotide and the polymer of nucleic acid, meaning made up of. While mRNA is similar to DNA, they have their distinct differences. While DNA contains a deoxyribose sugar, RNA contains ribose. Additionally, the structure is different. DNA is well known for its double helix structure, but RNA only has a single strand. Lastly, the bases are different. We know that the nucleotide bases in DNA are adenine, thymine, guanine, and cytosine. However, RNA does not contain thymine. Instead, it has a base called uracil. While they do differ greatly, they work together in this process of translation to form an RNA copy based off of the DNA template strand. The first step of translation is initiation, in which begins when RNA polymerase, an enzyme, binds to a promoter on the DNA. These promoters signals the beginning of a gene. RNA polymerase is used to make a strand of RNA that corresponds to the DNA template strand, or the strand in which it is translating from. Next, the phase of elongation begins. To build the new RNA strand from the 5' end to the 3' end, RNA polymerase adds free-floating nucleotides that complement the strand of DNA being transcribed, match with their bases. Lastly, termination begins. In this step, RNA polymerase recognizes the DNA sequence called a terminator that stops the transcription process as a whole. While the transcription process has been completed, mRNA processing now takes place until the next step in the central dogma. The mRNA contains sequences known as introns and exons. Both of these are groups of 3 bases like this. Introns are 3 base sequences that don't code for amino acids. Meanwhile, exons are 3 base sequences that do code for an amino acid. While mRNA is edited in this process, introns are cut out while the exons join together. Additionally, a cap, sequence, and tail are added to put it all together before the next step.
Next, in the central dogma, translation occurs in ribosomes within the cytoplasm of the cell. Translation is a process in which a ribosome reads the mRNA code and makes a protein that is able to perform a specific function. The ribosome is made up of 2 subunits, one large and one small. There are 2 sites within the large subunit, the P site containing polypeptide and A site containing amino acids. You should also understand that 3 mRNA bases in a row are named a codon, and each of them code for a specific amino acid. When the coding begins, the ribosomes read the start codon first. Each RNA code should begin with the codon AUG, which codes for the amino acid methionine and marks the beginning of a protein. After this, tRNA is used frequently. It is transfer RNA. Anticodons can be found in tRNA, and they complement the codons of mRNA. tRNA then brings amino acids to the ribosome. The amino acids are dropped off and are bonded together with a peptide bond between each amino acid. A polypeptide chain begins to form as the number of bonds increase and the chain elongates. Lastly, similarly to the start codon, a stop codon is translated to stop the reading. These codons are UAA, UAG, or UGA. Once all the proteins are created, a protein can be used for muscle tissue, transport, energy, movement, structure, chemical messengers, or immunity. While the entire process was carried out successfully in our video, what would happen if it didn't? Imagine if the amino acid sequence was incorrect because the RNA code was read wrong. If this happens, a mutation took place. In our situation, an insertion mutation took place. A new base was placed in the sequence. This altered the reading frame in every amino acid after the mutation was altered. Because this mutation alters the reading frame, it throws everything out of shape. Every codon behind the mutation is affected. The change in amino acid sequence caused a change in shape, thus a change in function. Therefore, because the function of the trait was affected, Pablo may be green instead of blue, and we all know Pablo is much better as a blue penguin than a green one.